Hello, in this video we will discuss about the physiological functions of the mitochondrial uncoupling protein. This is also known as the UCP. So in this way we will understand about the UCP2 and 3. So let's begin to understand what is the UCP2 and UCP3 in the mitochondria, inner mitochondria membrane. So the UCP2 and UCP3 are important potential target for treatment of aging. So basically aging and degenerative disease, diabetes, obesity, and uh, it is also the export fatty acid physiological weak and the export fatty acid physiological weak, but it can be and mild regulated uncoupling. They cause attenuates mitochondria reactive oxygen species production and protect against cellular damage. Also the UCP is also involved against the cellular damage, so that it is protection against the cellular damage and diminishes insulin secretion. In this way, it will inhibit the secretion of insulin. So let's begin to understand first of all what is the UCP and the function. So all these things, first of all, we should need to understand. Here is the cell, which that's contain nucleus, cytoplasm, plasma membrane or cell membrane, and the mitochondria and Golgi bodies. But we are focusing and targeting the mitochondria for understanding. So inner mitochondrial matrix and outer mitochondrial matrix are present within basically the inner mitochondrial membrane and outer. And the inner mitochondrial membrane contain five complexes. In this way, the ATP synthase enzyme, and in this way, the pump out the proton through oxidation reaction to reduction reaction will lead to produce ATP as well as UCP used for the influx of the proton into the inner mitochondrial matrix to produce heat. So this is the mitochondria. Let's begin to understand the further. And in this way, here is the outer matrix and outer mitochondrial membrane. And this is the inner mitochondrial membrane. And while here is the inner matrix. So in this way, the inner mitochondrial matrix is basically used for the electron transport chain and the oxidation reduction reaction due to the several protein complexes used for the proton pump out from the NADH and FADH2. And in this way, the NADH will be oxidized and remove the two electron and proton, and this electron will transmit further to the next complexes, will lead to outflux of the proton. In this way, the proton pump out. So this is the inner mitochondrial membrane, and this is the outer mitochondrial membrane, and the matrix outer and inner. So this is the NADH will be used to produce to lead to proton pump out for proton, and the electron transmit to the another complexes through a cytochrome C. But here is a ubiquinone or ubiquinone, which that will lead to transmit the electron to the other is the cytochrome C. So in this way, here you can see the proton pump out the 4 proton. In this way, when proton will be increased in the outer mitochondrial matrix will lead to decrease the pH, while inside will be increase the pH, mean more basic. So in this way, here is the oxidation to reduction when influx of the 4 proton into the ATP synthase enzyme lead to electron will be used, and proton and half molecule of oxygen will lead to produce water. It is known as the reduction reaction. But in this case, the proton also move inside through UCP2 to produce heat. While here is the inner matrix, which that contain a Krebs cycle, which that acetyl-CoA come from the glycolysis to pyruvate to convert the acetyl-CoA, and this will used and produce the NADH and FADH will use to proton pump out for the energy production and heat production. So here is the adipose tissue. Also lipolysis will occur. The will triacylglycerol convert into the glycerol and fatty acid in the hormone sensitive lipase enzyme presence. And in this way the fatty acid basically also lead to move out, the pump out to produce heat. In this way the fatty acid is also involved, and proton. So here let's begin to understand the thyroid gland is also involved for the trigger of the thermogenesis. Through T3 and T4, thyroxine and triiodothyronine hormone will produce from the thyroid gland from the thermogenic signal will lead to go enter into the blood circulatory system will target to the several tissue for non-shivering thermogenesis. In this way the target the DNA in the nucleus will transcribe the human sensitive lipase enzyme as well as into the DNA is also the UCP production of the mitochondria into the mitochondria. So the non-shivering thermogenesis will occur in the due to the production of heat from the UCP due to the UCP in the mitochondria. So ignore the insulin secretion, we will discuss now. So let's begin to understand the model of the physiological function catalyzed by UCP2 and UCP3. We will classify the different models of the cell, which that is contain the mitochondria and the cytoplasm and the plasma membrane. And in this way here is you can see these are several cell, which that is we will discuss the different condition. So in this way here is the mitochondria, and this mitochondria contain electron transport chain, ATP synthase enzyme as well as UCP, while this is the cytoplasm and the plasma membrane is here. When the lipolysis will occur through a hormone sensitive lipase enzyme, the triacylglycerol, which that produce the fatty acid, and this fatty acid go into the mitochondria and the fatty acyl CoA formation, and this way produce the proton from the NADH through acetyl CoA as well as NAFADH2, which that will be pumped out through electron transport chain. And when the proton will pump out through electron transport chain, this will use for the two way. One way is UCP and the other way is the ATP synthase enzyme. When influx of the proton in the ATP synthase enzyme to produce ATP, while when influx from the UCP to produce heat. So this is the 1A basal thermogenesis, the basal thermogenesis mechanism. But let's begin to understand the 1B inducible thermogenesis. So the inducible means the induced thermogenesis. In this way the fatty acid through a thermogenic signal which that will lead to produce the fatty acid, and this fatty acid basically can enter into the from the UCP directly into inside will produce heat, while on the other hand the electron transport chain produce proton and the ADP will be converted into the ATP due to the proton move inside, while the electron transport chain will produce the reactive oxygen species will also increase the UCP activity will produce the energy heat. While here is the third one is the M2, the attenuation of reactive oxygen species production. In this way the fatty acid will enter into the cell and go into the mitochondria to convert the fatty acyl CoA, and in this way you can see this is the proton will produce in the form of NADH will pump out through electron transport chain will use to the UCP and ATP synthase, and in this way the proton will be produce the heat as well as ATP. So after this next is we will discuss in the next model, the model 3, the regulation of insulin secretion, because the UCP is also involved for the regulation of insulin. How it is possible, let's begin to understand. So this is the mitochondria inside is contain electron transport chain, UCP and the ATP synthase, inner mitochondrial membrane. So this is the beta cell of the pancreas. So fatty acid when enter into the mitochondria, it convert into the fatty acyl CoA, and this fatty acyl CoA will produce proton in the form of NADH and FADH2. And in this way you can see this will pump out this proton from the electron transport chain via oxidation, will lead to also reactive oxygen species, will stimulate to proton pump in, and the fatty acid is also pump in. Inside, the influx of the fatty acid will lead to produce heat. So anyhow, the ADP will convert into the ATP decrease due to the proton is using for the UCP. So that when ATP will be decreased, means the insulin secretion will decrease. So remember. So this is the regulation of insulin secretion. On the other hand, here is the model for a fatty acid export, which that also produce heat. How it is possible? Let's begin to understand. This fatty acid will transport to fatty acyl CoA, and in this way the proton will also move inside, and this will lead to produce the NADH and FADH2, and in this way the ATP synthase enzyme use ADP will convert into the ATP, and on the other hand, the electron transport chain will pump out the proton and transmit the electron, while the pump outed proton will be used for the synthesis of ATP, or a reactive oxygen species will lead to trigger the UCP and will lead to outflux of the free fatty acid anion. This fatty acid anion will produce when the proton will be added. In this way, the fatty acid anion will be through UCP will outflux will lead to produce heat, and this is the fatty acid export will lead to cause thermogenesis, and in this way the heat will be produced. While here you can see the model 4b, fatty acid peroxide export. In this way, the fatty acid peroxidation will lead to outflux. In this way, the fatty acid go into the cell, and the fatty acyl CoA will be formed. And in this way, again, that same mechanism to increase the proton in the form of NADH and FADH2 will pump out through electron transport chain, and reactive oxygen species will trigger more the outflux of the peroxidation, the fatty acid peroxide export, and lead to produce more heat through UCP. And on the other hand, the ATP will also produce by this mechanism, and in this way you can see the proton will also move inside via again to enter in the form of the peroxidation will lead to basically the cycle will occur. In this way, you can see this is the model of the physiological function catalyzed by UCP2 and UCP3. And in this way, the basically it is a potential target for treatment of aging, degenerative disease, diabetes, and the obesity.